Cancer stem cell research has been at the forefront of cancer therapy research for understanding why cancer treatments aren't always 100% effective. To understand why traditional cancer treatments are not effective with this type of cancer cells, we must first understand what a stem cell really is. Stem cells are a group of undifferentiated pluripotent cells that are capable of dividing and renewing themselves for long periods of time. Stem cells are a key cell in the development of new differentiated tissue in embryonic growth and in tissue repair and growth in adults. Differentiation is regulated by different checkpoints and regulatory proteins in the cell cycle that ensures the replication process goes smoothly. Cancer occurs when a defective cell skips one or more of these checkpoints and does not receive death signals and thus divides rapidly into a tumor. Benign tumors remain in the tissue where the tumor forms, and malignant tumors spread to other regions of the body. The spread to other tissues is known as metastasis, and it is attributed to the presence of cancer stem cells, which we'll be discussing in detail later. Before being able to treat a patient, we must first ensure the presence of cancer and identify the type of cancer present. A commonly used approach to identify the presence of cancer is RNA sequencing. Cells naturally accumulate a number of mutations in their DNA. However, only a few specific mutations result in missense or nonsense defects, some of which include a single base substitution in codon 12 that causes glycine to become valine and is linked to bladder carcinoma oncogene, the translocation between chromosome 9 and chromosome 22 in chronic myeloid leukemia, and the addition of extra DNA sequences acquired from viruses. By sequencing the genome of suspected cancerous cells or tumors, we can look for these specific known genetic defects and thus correctly identify the type of cancer present and find if there are any cancer stem cells present in the tumor. There are many different types of cancer treatments, and they are all used to kill, remove, or limit the growth of the tumor. Some examples include surgery, chemotherapy, radiation therapy, and targeted drug therapy. The type of treatment is dependent on the type and stage of cancer. However, these treatments are not 100% effective. An analogy for the treatments would be a weed treatment. No matter how short you cut the stalks, if the root is not killed, the weed will grow back. Tumors can survive because of cancer stem cells in the tumor. Cancer stem cells are able to replicate and divide to produce more copies of itself or differentiate into other types of rapidly proliferating tumor cells.